Alright, since we've got 2 days next week,
Alright.
might as well get started on a whole nother chapter.
Yes.
Well, you can disagree all you want, I'm not going to change anything. Okay, now, don't forget you do have that DNA project that is coming up. So, hopefully you've at least thought about that. We really should have been through the chapter on DNA by now, but because of all the interruptions and things, as far as snow days and all that kind of stuff, we're about a week behind. So, yeah, that's one reason. That's one reason we're definitely going to get started today. But we will be learning about DNA in this chapter and the structure. So after next week, you'll have a little bit better idea on the structure of DNA from class, and that should help you in building your molecules. But of course, you still have a couple weeks after we come back from Christmas to get those done too, because they're not due until was it the 13th, I think. It's one of those. It's either the 13th or the 15th. I'll take a look at that. But today we're going to start on Unit 8. For this unit, to change things just slightly, I'm going to hand out this packet, and this packet has basically all the handouts for the chapter. So all the worksheets that you'll do are already in here, unless I decide to add something. So I won't have to hand those out every day. You'll just already have them. Okay, so the first page is your study guide. You can look over that, hang on to that for the review. The second page, or I guess on the back of the study guide, is your word list. And you can see not near as many words this time. So we should have lots of good grades on this, because there's not that many to remember. I definitely considered having this test on Tuesday, or the quiz over the vocab words on Tuesday. But we'll push it back till after Christmas.
All right.
So the Wednesday after Christmas, which would make that the 7th. So January 7th will be the quiz over these words. I'm sure most of you will look at those on January 6th, but at least you know when it's coming. All right. Today we need to start talking about we're going to get kind of a preview towards the discovery of DNA. We won't talk about DNA till tomorrow. On Monday, Captain Literal. Okay. Listen up, please. Mendel had kind of a tough job when he was looking at all of his plants, because he figured out all the stuff that we learned, and we were able to learn that fairly well. But you guys have the advantage of knowing about chromosomes ahead of time. We talked about chromosomes and that kind of stuff when we looked over cells and their structure. Mendel didn't know anything about chromosomes. So imagine trying to figure out all those different things without knowing how chromosomes are set up, that there is DNA, any of that stuff. He had no idea. And it wasn't until the 1900s, the early 1900s, that we actually figured out how genes were being passed on, or what they were being passed on with, anyway. So Mendel figured out kind of how heredity worked, as far as, oh, you've got these alleles and they get passed on, and depending on which ones you get, that determines the traits that you have. But he didn't really know what the mechanism was. Why were those things getting passed on? What was actually going on in the cells that you got these traits? Okay? So this guy here is a guy named Fred Griffith. And he was doing some studies in 1928. Okay, now comparably, Mendel was doing his experiments in about the 1850s, so right before the American Civil War. So it was quite a few years after Mendel figured out all that stuff about heredity that Griffith actually came along and started to figure out at least the initial stages of how things were being passed on, the mechanisms. Okay. Now, what Griffith was looking at, next slide here, was different strains of bacteria that cause disease in mice. Okay, so these were bacteria. And he had two different strains. He had one strain that we call type R, that when you injected that bacteria in the mouse, nothing really happened. Okay? Didn't cause any disease in the mouse. Everything was fine. He had another type called type S, that when you injected into the mouse, it killed the mouse. So, mouse died. Now, the experiment that Griffith did is he took some of the type S and he heat killed them. He basically heated them up until it killed the bacteria cells, and then he injected the dead cells into the mouse. Okay? And the mouse lived because the cells were dead. Now, people had done that before to show, okay, bacteria are living things. If you kill them, the cells will not infect you. But what he did that was kind of neat is he took some of those heat-killed S cells, the ones that were dead, and he injected them, but he mixed in the type R, the ones that didn't kill the mouse, the ones that were not harmful to the mouse. And what happened was the mouse died. So he realized that there was something going on. There was some sort of material that was being passed from the dead S cells, the lethal ones, the ones that are supposed to kill the mouse, but couldn't because they were dead. There was something that was getting passed into those R cells that made them kill the mouse. Okay? He called this transformation. Okay, and well, this transformation might not be quite as exciting as watching a Camaro turn into an 18-foot tall talking robot. It's still kind of interesting, the fact that you have, a cell that is not supposed to be harmful to the mouse being mixed with, dead cells, and they're somehow able to get some sort of material that causes them to then become lethal. So this was the first kind of real evidence that there was some sort of actual material, some sort of thing that could be passed from one animal to another that was involved with the genetics. Here's what happened to my list of stuff. Oh, well, we'll press on. Hey, this guy, his name is Oswald Avery. Too many people being named Oswald anymore. Maybe that'll come back someday. And Avery took Griffith's experiments, but he took them one step further. he was kind of able to isolate different materials of the bacteria and figure out what was actually being passed that caused those bacteria to become lethal. So I don't know if you can see this very well here, but we know that there's 4 main organic molecules, right? You got proteins, you got sugars, you got lipids, and you got nucleic acids. So they kind of, it made sense. One of those types of materials is going to have to be what's being passed from one bacteria to another. So he basically did the same experiment that Griffith did, but instead of taking the whole dead cell and putting it in with the living bacteria cells, he just put parts of the cell. And what he found is when he did it with lipids, sugars, polysaccharide is another name for a sugar, and proteins, nothing happened. So he realized, well, those can't be the things that are actually making the cells kill the mice. When he did it with nucleic acids, though, that's when the cells turn lethal. So the transformation only happened when you had nucleic acids that were able to be transferred into the living cells that would then kill the mice. So what Avery did is he proved that nucleic acids carry genetic information. Okay? But the problem is a nucleic acid is actually made up of a DNA strand, but there's proteins that DNA strand is wrapped around. And so your basic nucleic acid is going to have DNA and its own proteins. And there was a lot of scientists that were still thinking, well, it must be the proteins that the DNA is wound around that actually cause the genetic information. And so even though Avery was able to show, well, these things don't, have anything to do with how heredity works, they still didn't know, was it the actual DNA part of the nucleic acids or the proteins they were all wound around. And so it took another group of scientists to actually figure out what was going on there. Now, I want to take a minute here and talk a little bit about viruses, because we're going to have to understand what a virus is to understand the next experiment. There's lots of different types of viruses, and they have lots of different shapes. The viruses are pretty simple organisms, okay? They have, and in fact, some scientists even don't even consider them to be living, but they have kind of an ornate structure on the outside with special proteins, and those things are going to be able to help that virus attach to the host cell, but they're fairly specific. That's why if you take a virus that infects plants and you inject it into yourself, it's not going to do anything to you because, excuse me, that virus is set up to attack plant cells, not your cells. But there are some that are, going to work on us. And so these are all examples of ones that would be harmful to humans as far as their shape goes. But the overall shape of a virus is not that important to the actual structure, that you basically have this outside part called the capsid, and inside you have some either DNA or RNA. Those are just 2 different types of nucleic acids. Now, if you were to get some sort of virus, say the influenza virus, what happens is that virus gets inside of you, it attaches to the outside of one of your cells, and it shoots its DNA or RNA into your cells. And then it uses your cells to actually make more of itself. So it's kind of like, that's why we call things computer viruses, after the way the viruses work. They inject themselves into the host, it lets the host cell replicate it, and then those things can go on and infect new cells. Right? So as scientists started to learn about viruses and how they work, they realized, well, this is pretty simple. We got this thing, it shoots its DNA into a cell. We can use that to study how the DNA affects living cells and whether or not it's the nucleic acid part or the protein part that's actually involved in heredity. This is one type of virus called a bacteriophage. I think bacteriophages are kind of the coolest looking type of virus. Their setup is slightly different. They've got these kind of hook things on the bottom that they use to grab onto cells, and then they inject their DNA inside. It does look kind of like a squid. It's cute. There we go. But even though these are kind of cool looking viruses, we don't worry about them because all bacteriophages, as the name implies, all they infect are bacteria cells. So a bacteriophage just isn't set up to attack human cells. There's nothing it can do. But bacteriophages are used all the time to study how simple genetics work because bacteria cells are simple and easy to study, and the bacteriophages, how they interact with them, are simple and easy to study. So we can use that to kind of gain some insights to how DNA works. So here's another drawing of a bacteriophage, so you can kind of see the different parts to it. So it's got the tail fibers and the sheath. This is a helical protein. But it's pretty simple. This part right here is just the carrier for the DNA. Oops. I think I lost a picture here. Okay, I'll draw it. Not a problem. You guys like my drawing skills, right? So, here's a bacteria cell. Now we know bacteria cells are pretty simple. They don't really have much. Yes, they have a cell wall, so we'll throw the cell wall on there. They've got some ribosomes, and then they've got some DNA. But bacteria cells are neat in the fact that their DNA is in a complete circle. Yeah, there's a few other things that go along with bacteria cells, but that's really the most important stuff. So a bacteriophage, when it comes along, oh, I get to draw this. This will be fun. Yeah, that's not too bad. Okay. Just keep that off to the side in case I screw this one up. Make it a little smaller. When a bacteriophage comes along, now they don't move. They just kind of float along. See, I hate it when it does that. There we go. Okay, they just float along. But if they happen to bump into a bacteria cell, those tail fibers will grab onto it, and then it will shoot its DNA into the bacteria cell. And that DNA is floating around. And when this cell replicates, when this bacteria cell is going to go through binary fission and make another one of itself, because this DNA is in here, the bacteria cell can't tell what part of the DNA is its and what part is from the virus, and it makes a copy of that too. And then that DNA actually is able to force the cell to make more copies of itself. And so you get all of these copies of viruses inside it until the cell is so full of them that the cell explodes and all these viruses escape. Some escape better than others. Okay? So that's the basic life cycle of a virus. Attach, inject your DNA, let the cell make more of the virus for you, and then the cell, we say, lyses and explodes, and those viruses can spread to other cells and infect other cells. Okay? Now, because the DNA, during this life cycle, because the DNA becomes separate from the rest of the virus, it's easy to take a look at how those things are going to affect spreading. So, let's see, I gotta look here to make sure I get these people's names right. This is Alfred Hershey and then Martha Chase. And in 1952, they performed an experiment to really definitively lock down nucleic acids as being carriers of genes. So Hershey and Chase. So you know which is which. Yeah. Now what they did is they used radioactive elements to kind of track how the DNA got passed into the cell and what actually caused the cell to go through, what we call transformation. So here's the basic experiment that they did. Okay, in one case, they took this, these bacteriophages and the bacteria cell. We've got the DNA, everything. And they took it and they put it in with radioactive particles so that, and I believe in this case it was sulfur, so that it would get incorporated into the proteins. Okay? So right here, the type of radioactive particle that they chose was so that it would end up in the proteins. So they put bacteria, they put the bacteriophages, they put this radioactive material in that they knew would end up in the protein. They let it all go, and then they stuck it in a blender and blended it all up, put it in a centrifuge, and what the centrifuge does is it separates things out. So down here is going to be all the bacteria cells. Up here is going to be all the empty viruses. Okay? And what they noticed is when they tried to set it up so that the radioactivity would end up in the proteins, all of the radioactivity was where the empty viruses were. So what that meant was the stuff that was getting put into the cells didn't really have any proteins involved in it. Then they did the same experiment, except they changed their radioactive mixture right here, so this would end up in the DNA. Did the same experiment, and when they looked for the radioactivity, in this case, they realized the radioactivity was actually inside the cells. So what this experiment did is it definitively proved that the thing that was causing the cells to replicate the viruses and stuff was all about the DNA. It didn't have anything to do with the proteins. Because all the DNA stuff ended up in the cells. The stuff about the proteins was on the outside where it didn't have anything to do with the replication of the viruses. Okay? So those were the basic experiments that led up to us realizing, hey, it's all about the DNA. The passing of genes from parent to offspring, or from bacteria to bacteria, or from virus to bacteria, is all about DNA. It has nothing to do with the proteins. And then people started going, okay, well, if we know the DNA is what passes genes on from parents to offspring, how is that thing set up? What is the structure of the DNA? how are the genes actually arranged on? So that's what we'll have to start talking about tomorrow. So tomorrow we'll talk about the structure of DNA. We'll talk about Watson and Crick and some of the kind of underhanded things that they did. And one of the least known, but should be most famous scientists in all of biology. No, Rosalind Franklin. So here's your question of the day. Okay, we don't actually have any homework for tomorrow, so you get the weekend off tonight. I do need your homework cards, so go ahead and pass those to the end of your row of flat days.